Hello, everyone. Good afternoon. It's such a pleasure to see you all today. My goodness, into the third year of a global pandemic, and here we are on another virtual meeting. And I so appreciate the time you are taking out of your busy schedules to join us today to hear about our multiple myeloma program at the Princess Margaret. My name is Mio Yamashita. I'm the president and CEO of the Princess Margaret Cancer Foundation. I joined our foundation 7 months ago, and I should probably say rejoined the Princess Margaret. I actually worked here much earlier in my career, about 20 years ago. March is Multiple Myeloma Awareness Month, and you're going to hear from some wonderful speakers today. Shortly, you'll hear from Dr. Keith Stewart, who's also back relatively recently to the Princess Margaret. He was over at the Mayo Clinic. He's going to give us an overview of the Multiple Myeloma Program. But Keith and I are both so happy to be home, if you will, back in Toronto and back at the Princess Margaret at University Health Network. Dr. Keith Stewart is our Vice President of Cancer at University Health Network and the Princess Margaret. He'll be introducing 2 of his research colleagues. You'll also hear from a great supporter, and you'll hear about his family's experience with cancer and his family's involvement in one of our events. And then we'll wrap up with Greg Lichty, who's an Associate Vice President over at the Foundation. So thank you again for joining us today. Just a minor housekeeping item. We welcome your active participation in our webinar today. If you so feel inclined, please feel free to insert your questions directly into the Q&A function in our Zoom chat. And Dr. Keith Stewart, I will now turn it over to you. Thank you very much.
Thank you, Mio. It's an honor today to be here to talk about our myeloma program, which is where I started at University Health Network before going south to the United States and then coming back again in the midst of the pandemic. So it's just great to be talking about the future now instead of COVID-19 in every meeting. Our myeloma program is one of the largest in the world. We see between 400 and 500 new myeloma patients every year. This is supported by 8 hematologists, and you'll hear from Dr. Reece later, a very large supporting team of international fellows, clinical research coordinators, nurse practitioners, nurses, etc. We conduct over 200 autologous blood stem cell transplants in this program. And so I've just lost my screen. Excuse me 1 second. Our electronic health record just popped up. There, it's gone away again. Thank you, but sorry about that. And in the last year, as you'll hear from Dr. Chen, we've started one of the first in Canada to deliver what we're now calling immune intervention. And this is harnessing our own T cells to target the cancer with great success. Each year, we conduct over 40 clinical trials. And I'm proud to say that there's not a single drug that's been developed in myeloma over the past 20 years, and there are many new drugs that have been developed, that Princess Margaret has not played a very prominent role in their development, thanks to our extraordinary clinical trials program. It's a source of great pride to us that not only do we have a very active program, but the percentage of patients that go on our clinical trials is extremely high compared to our peers in Canada, the United States, and in the United Kingdom as examples. Over 20% of our myeloma patients participate in a clinical trial. This is almost 4 times as high as you would see in a United States equivalent center. We're also proud of the fact that of those patients who do go on trials, they're from incredibly ethnically mixed background, and the diversity of accrual to our trials is also a source of strength and pride. We have continued to focus on developing new therapies. I've mentioned already chimeric antigen receptor T cells. This is a situation where we take T cells from the patient themselves and we re-educate those cells, as Dr. Chen will explain, to both find the myeloma cell, but also bring their T cell friends to the party as very effective killers of myeloma, and really extraordinary results, as you'll see. In fact, I often tell people I've witnessed two medical miracles in my career. And the first was the discovery of thalidomide as being active in multiple myeloma, which was an extraordinary finding, which completely changed how we treat the disease. And the second has been chimeric antigen receptor T cells, which are just almost amazing to watch in action. In addition to our clinical trials, we've been able to, at Princess Margaret, Dr. Reece has stimulated a group across Canada, Canadian Myeloma Research Group, that has extended our reach across the country with multiple partners conducting practice-changing clinical studies. Our research team is also strong. Dr. Tiedemann and Trudell in particular running basic science laboratories, along with Dr. Trevor Pugh. We're focused on a number of things, but these include the development of new small molecules for treating myeloma, new drugs, the use of cells beyond just T cells, natural killer cells, another component of the immune system, the use of what we call theranostics, using antibody therapies to deliver radiation to the tumor so we can both see the tumor and kill it at the same time. We're focused on trying to get rid of painful bone marrows. We have introduced conscious sedation to make bone marrow experience more pleasant, but we think over time we'll be able to completely replace that using simple blood tests with a process we call liquid biopsy, which will remove the need to do continuously bone marrow aspirates, which can be painful if done improperly. We're also continuing to explore better molecular targeting with a more deep understanding of the molecular makeup of each individual patient's tumor, moving into whole genome sequencing, which used to be astronomically expensive, but now is affordable on a reasonable time frame and scale. And we hope to introduce this in future years, and are working hard now to implement that technology. Now, to that end, I did want to thank the foundation and our philanthropic support. Every year, we rely on the incredible generosity of our donors to keep this program as one of the leading programs in the world. They support our researchers, our scientists, our clinical research, and our clinicians, and we're forever grateful for the patients and the families who participate in our clinical studies, without which we could not make these advances. We're particularly pleased today to announce that the Princess Margaret has recently received an extraordinary donation from Helga and Antonio de Gasperis, supporting our multiple myeloma program. This incredible gift will fund a variety of purposes, including early detection through liquid biopsy and prevention, precision medicine for our multiple myeloma patients, and will provide access to novel therapies and clinical trials. Additionally, this gift will start a Grand Challenges program, funding the best research with the most chance of very quickly changing how we treat this disease. It will kickstart the most promising clinical trials and allow us to adopt the most advanced technologies faster than we were previously able to do so. This will be through the establishment of the Helga and Antonio de Gasperis Grand Challenge clinical trials in myeloma. So we're very grateful to the family for their outstanding commitment to transform myeloma research and care. And with that, I'd like to turn it back to you, Mio.
Thank you so much. And I want to echo your wonderful words of gratitude to the de Gasperis family and to all of our supporters. We really are able to be a world-class multiple myeloma program because of supporters like you. And Keith, I'm in awe hearing about some of the future advances that are occurring in multiple myeloma, including the possible early detection through liquid biopsy. Keith, we've got a bit of ping pong going because I'm actually going to pass it back to you to introduce our clinical colleagues. And that's because I don't want to mispronounce some of the clinical terms. So back to you, Dr. Stewart.
So we'll move on to the more formal part of the agenda. And I'm very pleased to introduce you to Dr. Donna Reese. Donna is the director of our multiple myeloma program. and a clinical investigator at the Princess Margaret Cancer Center. As a clinical investigator, Dr. Reece's research interests include hematopoietic stem cell transplantation, plasma cell dyscrasias, and the use of real-world data in her decision-making. She's developing enhanced capabilities for risk assessment in multiple myeloma and is using high-end analysis tools to allow deep resolution and molecular monitoring of multiple myeloma progression. Dr. Reece, over to you.
Thank you very much. It's a real treat to be here after several years of quietude. We have had these webinars in the past, and today I hope to bring you around to what we've been doing during the pandemic, or I should say in spite of the pandemic, and provide a taste of the range of research that we're doing at Princess Margaret. Next slide, please. Now, I think it's fair to say there are three components of our program. There's the laboratory research program. We also have a robust database that I'll describe a bit in a moment. But a real thrust of my talk will be our clinical research program that involves patient care. Next slide, please. The ultimate goal of all of these components is to deliver better care for myeloma patients with the ultimate goal of improving longevity as we work towards a cure. Next slide, please. I'll give you an overview of our research program. Our infrastructure consists, as you've heard, of 8 MDs. We have a research manager, 15 coordinators, and 5 research nurses. And I will say off the bat that Princess Margaret and our healthcare system provides excellent care for the standard treatments, but all of this infrastructure relies on the funds we can bring in for these trials and increasingly on philanthropy. We have excellent clinical trial capabilities. We're opening 10 to 15 new trials per year. We have 88 active trials, and we are able to do the full spectrum of trials with new drugs and combinations, what we call investigator-initiated trials. Those are ones we write rather than them coming from pharma. And as you have heard and will hear, we're focusing on immunotherapeutic platforms, immunotherapy with so-called conjugated antibodies, BiTEs, bispecific T-cell engagers, and as you'll hear from Dr. Chen, CAR T cells. These encompass only really 2 of the many types of immunotherapy. But whereas this was really a remote, almost science fiction goal when I came here over 20 years ago, the promise of immunotherapy is touching the lives of many of our patients in the last couple of years. Next slide, please. Our laboratory research program, I think, is what gives our clinical program real depth. We have 2 clinical scientists, clinician scientists, Dr. Suzanne Trudell, and I don't have time to go through all the work she and Dr. Tiedemann have done, but to echo what Dr. Stewart has said, Dr. Trudell has identified targets for drug development. She's has an interest in furthering immunotherapy and has really been instrumental in bringing the new drug belamaf to fruition, involved in new ways to measure the myeloma protein other than the conventional serum protein electrophoresis, and also involved in a sophisticated technique, single cell analysis of the molecular programs that are associated with myeloma's progression as it tends to become more aggressive with time. Dr. Tiedemann made a landmark discovery of myeloma biology in the past and is leveraging this to do a spectrum of research, including developing antibodies against these precursor or feeder cells that can give rise to myeloma after what appears to be successful therapy. We also have a wide range of laboratory personnel that include postdocs, lab technicians, and we're a training ground for future researchers in myeloma, graduate students who are working towards a master's or PhD. Next slide, please. Our myeloma database originated many years ago. Dr. Chen was involved as we were trying to keep track of the outcomes of our patients. We now have one PhD manager, 2 coordinators, and we moved from being a single site generating research results from this database to a national expansion. As Dr. Stewart mentioned previously, we have 17 sites contributing data on myeloma. I've just shown you at the bottom circle there, we have over 8,000 patients from these 17 sites, and the blue 34% there is our contribution with Princess Margaret data, with robust contributions from Ottawa, Alberta, and other evolving centers. What can we do with this database? We can look at real-world evidence, benchmark where we've been, define gaps where we need to go. And I'm happy to say we have finally caught the attention of our provincial funders. It's an organization called CADITH who ultimately recommends whether the provinces fund a new regimen or not. We've been knocking at their door for years, and they are now looking at our data to see what makes sense in terms of getting new drugs to patients. And we are evolving interactions with Health Canada. Like the FDA, Health Canada is starting to look at real-world evidence in the process of new drug approval. Next slide, please. What has Philanthropy accomplished? As I've said, we have good provincial funds for delivering basic care, but philanthropy is needed for really to subsidize much of our other work. Philanthropy has specifically funded the basic research of Drs. Trudell and Dr. Tiedemann, and some very exciting work done by Dr. Sofak with recent presentations at the large ASH meetings. Our database includes our own 4,000 patients centered at Princess Margaret, which we routinely assess to see how we're doing with different treatments. And then I'll talk more about our clinical research program in the next few minutes. Next slide, please. I should also add that it's a different matter to take this basic research and actually get it into the clinics. And due to philanthropy specifically, we've been able to hire a correlative lab manager who has been successful in getting the 38 gene panel, which I'll speak to in a minute, as a clinical test that can be ordered with the cost subsidized by philanthropy. We have another PhD who has been hired as a translational researcher. She was trained in Dr. Trudell's lab, all to help in this bridge of bench to bedside. Next slide, please. The database is funded by philanthropy. There is no money in the healthcare system to track this sort of real-world evidence, and we use this evidence for academic results and, as I said, as benchmark for funders and for other enterprises. Next slide, please. Now, for the clinical research program, we do rely on philanthropy to fund selected coordinator salaries. And in contrast to 15 years ago, we do rely on donated funds to help subsidize our ability to do what we call investigator-initiated trials. The deal with these trials is we get the drugs from pharma, but we don't get much funding to support the research nurses, the coordinator activities for reporting to Health Canada, et cetera. But the trials that we have been able to bring to patients with this philanthropic back for and after stem cell.
Donna, we're losing you. I wonder if you might turn your camera off to see if that helps.
We did
Donna, I'm sorry, we seem to be losing you. We seem to have lost Donna, so I think her bandwidth exhausted itself there.
Am I back?
You're back, Donna. Thank you. We lost you sort of halfway through the last slide.
I have no idea what happened, but I'm glad to come back. So I'll leave my video off in case that helps. If I should run through the trials again, these are important trials that have brought drugs to patients with monoclonal antibodies before and after stem cell transplant, which is the wave of the future, and it will be a while before we receive provincial funding. These donated funds have been used to support conjugated monoclonal antibodies with pomalidomide and a very effective daratumumab 4-drug quadruplet for relapsed refractory myeloma and a carfilzomab combination for relapsed refractory myeloma. Next slide, please. Dr. Chen's going to talk more about CAR T cells, but I thought a complementary program merits mention. This is the strategy of the BiTEs, and these are a permutation of bringing a killing T lymphocyte from a patient to attack a marker on the surface of a myeloma cell. And I'm happy to say that we have access and trials with all the major BiTEs from the pharma companies listed on this slide, and a schema that shows that a T cell can be engineered, can be attached via this antibody, which has one arm sticking on the T cell, the other arm on the tumor plasma cell, so that the T cell can directly kill this malignant cell. And we are going to be able to move this type of strategy into first-line therapy with a cooperative trial with the European Myeloma Network. As Dr. Stewart mentioned, you can also use this strategy of a bridging therapeutic antibody to bring not a T cell, but a different cell, an NK cell, to the myeloma cell and also result in killing. And I think one of the very attractive aspects of this technology is that there may not be the fever or chills that can be seen with the BiTEs. There's no CRS. Next slide, please. In terms of assessment of patient risk and measure of tumor burden, a couple of highlights are shown on this slide. Doctors Pugh and Trudell have worked on a 38-gene panel that can be performed in the blood or on bone marrow, again, maybe replacing the need for repetitive bone marrow biopsies. And using this analysis, one can better assess which patients have higher risk disease and may need different strategies, and who may have more indolent biology. We'd love to move beyond the era of serum protein electrophoresis, measuring the monoclonal antibody to detect the disease burden. MRD, or minimal residual disease in the bone marrow, can detect a very rare residual tumor cell, and the wave of the future will be to escalate or de-escalate therapy depending if low numbers of these residual cells are present. And I've already mentioned a new blood test to measure the M protein. This was a recent publication on this mass spec technology called EasyM. All of these were made possible by Philanthropy and accelerated in the clinic by our scientific coordinators that I mentioned previously. Next slide, please. What about patient care? I always say that clinical research is predicated on meticulous patient care, and our donations have helped us hire a second nurse practitioner to be up full-time in the daycare unit to manage newly diagnosed patients, to manage those who are receiving monoclonal antibodies or complex care. They can assess and treat acutely ill patients and avoid the need to go to the emergency room. And they're really experts in symptom management and palliative care. You've heard about conscious sedation. This is really a very important way to improve the patient experience. And it allows, in addition, for us to take a few more cc's or milliliters of marrow for the newer tests such as MRD, as well as for research if the patient provides permission. Next slide, please. So future directions is to collect these different assays and techniques to create a myeloma rapid diagnostic program. The pieces are falling into place. I've talked about the tests for myeloma biology. We are now doing PET CT scans and diagnosis for all patients and are replacing the skeletal survey with whole body CT. I haven't had a chance to talk about the variety of non-interventional studies that look at key aspects such as quality of life, the impact of frailty on outcomes, caregiver burden, and a better way to assess risk of side effects and toxicity with stem cell transplant. And finally, the continued optimization of immunotherapy to work towards a cure of this disease. Next slide, please. So I had meant to also include a sincere thank you to all our donors, including the DeGasperis family, for their significant donation to the myeloma cause. Thank you.
Thank you so much, Dr. Reece. We'll now move on to, and we'll have lots of time for question and answer with Dr. Reece as the afternoon moves on. I'd like to now introduce our next speaker, who is Dr. Christine Chen. Christine is actually responsible for getting me back to Toronto. She sent me a text in the fall of 2019 asking if I'd be interested in coming back, so you can blame her or thank her. Christine is the medical director of the Autologous Stem Cell Transplant Program and the clinical lead and quality director for our immune effector cell therapy program at the Princess Margaret. In this role, she provides oversight both for standard of care and research into the cell therapy delivery and processes. As a clinical investigator, Chris's interests are in the development of novel approaches for the treatment of myeloma and a related disease, chronic lymphocytic leukemia, focusing on cell-based immunotherapies. So with that, Chris, I'll hand it over to you.
Thanks, Keith. And I am actually quite happy you're back in town. It harks back on the old days with you as my mentor and Donna just freshly on staff with us 20 years ago. So I was asked to talk a bit about cell therapy in myeloma. And cell-based therapy is essentially the use of living cells as part of treatment for cancer. And although, sorry, if you stay back on the previous slide, and although we talk about the promise of CAR T cells, which is a relatively new innovation in immunotherapy, I don't think we should forget that we've been exploiting the use of stem cells for over 30 years with regards to autologous stem cell transplant, which has been a cornerstone of frontline treatment for myeloma for the last 30 years. So I am going to touch a bit about stem cells as well as CAR T cells. So next slide. So many of you are very familiar with stem cell transplant, and I'm only going to show one slide to get everyone on the same page. Hematopoietic or blood stem cells have the ability, they're progenitor cells or mother cells that have the ability to differentiate into all the different components of blood, red cells, platelets, white cells. And we're able to collect these cells from the patient's blood, freeze and store these cells, and it allows us to administer high-dose chemotherapy or sometimes radiation as well to patients with myeloma to try to ablate or kill the myeloma that resides in the bone marrow. And unfortunately, the bone marrow also the normal bone marrow can also be damaged, and hence the infusion of the frozen stem cells, which are thawed, is a way to rescue the bone marrow and allow for normal production of blood. Next slide. So stem cell transplant is highly effective. We know that with many advances made, we now, combining transplant with novel therapies, we can achieve very deep and durable responses that can last for years. We performed our first auto transplant, so autologous, meaning from the patient, him or herself, in 1986. And we are the largest autologous transplant program in Canada, performing 300 to 350 auto transplants per year for diseases including myeloma as our primary indication, but also lymphoma and other diseases. It's hard to believe that it's been 7 years since we opened our outpatient transplant program that allows patients to undergo transplant from the comfort of their own homes with daily visits to our autologous day hospital that you can see here in the image. I wanted to highlight a bit of the some of the transplant advances made in myeloma at Princess Margaret and what we are doing now to further this already quite effective treatment. We are looking at highly effective induction therapies as well as maintenance therapy. Donna briefly mentioned the use of monoclonal antibodies both before and after transplant that may help to sustain responses at a deep molecular level. Using Suzanne Trudell's 38 gene panel that you already heard about from Donna, we're able to identify high-risk patients who are then funneled towards 2 transplants or tandem transplants, one after another, followed by very careful surveillance using our minimal residual disease flow testing, as you know, also highly supported through Philanthropy. We also are developing novel regimens for salvage transplants. These are transplants performed years after the first transplant. We will repeat the transplant using different induction or maintenance regimens. And then there are a variety of other initiatives that I just wanted to highlight. The frailty assessments that help us to optimize the patient path through transplant for those who are most vulnerable, and even encouraging the development of education modules online for patients, caregivers, and their families, as this is a very complex therapy that requires some hand-holding to get through. So we've recently completed our modules, again with heavy support from Philanthropy, and these can be available and viewable on the pmcancerclasses.ca. And I'll leave it at that. There are many other initiatives over the last 30 years that have helped to advance transplant. Next slide. But you've already heard about exciting chimeric antigen receptor T-cell therapy. So what is a CAR? Well, the image on the left shows a CAR that is a molecule, well, a structure that spans from the inside of a T-cell that's on the lower part of this slide, with the part that's extending outside of the T-cell having the ability to recognize and bind to a marker on tumor cells. And currently in myeloma, the primary marker is BCMA. So many of you will have heard about this ubiquitous marker seen on myeloma cells. Once the CAR, the external part of it, binds to the tumor cell, it sends a signal down into the T cell, which activates this T cell. And this T cell, now activated, has the ability to destroy the cancer cell that it is bound to, as well as release chemicals like cytokines that can recruit other immune cells to further cause death of cancer cells. This is a highly effective treatment with a single infusion, though you can imagine that the activation of numerous cells that are releasing these chemicals can play havoc with with symptoms such as fevers, shortness of breath, neurotoxicity, and risk of infections. And therefore, this is a challenging treatment for patients, to say the least. Next slide. and challenging also to usher patients through this very complex and intense therapy. And you can see that on the image on the left outlines the process for CAR T cell therapy that somewhat mimics that of auto transplant that I showed you earlier, where patients have instead of stem cells being collected from their blood, they have T cells collected. And these T cells then outside of the body are genetically engineered to express that chimeric antigen receptor that allows those T cells to recognize the cancer tumor marker. These cells then are multiplied into millions of cells and then frozen and returned to the patient. We thaw them and infuse them as a living drug. And you can see that this whole process will take a village, not just the clinical team with physicians, nurses, pharmacists, and trainees, but also coordinators who usher patients through the whole pathway, the cell therapy lab that will handle and manipulate the cells, as well as the apheresis lab that collects the cells, and numerous other staff, all of whom require training and a steep learning curve for developing expertise in all these steps of handling both the cells and the patient. And not to be belittled is the need to follow these patients with long-term data, which is mandated by Health Canada for a minimum of 15 years. Next slide. So is all this effort worth it? Well, you can see that on this slide, I've simply shown the overall responses of virtually 100%, 97.8%, 9% of patients receiving one of the BCMA CAR T cells, cilta-cel, will respond. And remember, these are heavily pretreated patients in this early phase study. These patients have received a median of 6 prior lines with no real standard of care options left. And yet they respond uniformly, with responses deepening over time, even without ongoing treatment. Remember, this is a single infusion of CAR T cells, with over 90% achieving MRD or deep molecular remissions, and most patients not progressing at 2 years, with excellent quality of life, with no ongoing treatment. This is probably the first time in for many of these patients where they have had any significant period where they are off therapy for myeloma. Next slide. So on this slide, I wanted to highlight a few just glimpses into advancing CAR T cell therapy at Princess Margaret, some of the initiatives we are taking. So next slide or forward. We are involved with various CAR T trials in myeloma using autologous products, ide-cel, cilta-cel, CT053 trials. So all using slightly different cell doses and management approaches to try to hone efficacy and minimize toxicity. Next. I showed you previously the amazing results of CAR T in heavily pretreated patients who are often refractory to all treatment available. You can imagine how moving CAR T earlier into the front line may potentially even lead patients to cure. So we are participating in the CARTITUDE-5 trial, that is a frontline CAR T trial. Next. We're also shifting CAR T into the outpatient setting. This is intensive treatment where the use of the ICU for cytokine-related complications may be required, and therefore it's a major. There are major accolades to our clinical team for allowing us to move this type of intense treatment into the outpatient setting. Next. I talked a bit, and so did Donna, about assessing patients for their frailty and identifying vulnerabilities. We are leading a trial primarily through Ontario Health and our center in assessing frailty using a variety of tools. The image here is that of grip strength measuring, which is a very good and simple measure of overall fitness. Next slide, or next. One of the challenges with CAR T-cell therapy when we use autologous T-cells, so from the patient, him or herself, is the time it may take to manufacture cells that are expressing the CAR receptor. And so we are investigating alternative approaches such as allogeneic CAR Ts, where we take T-cells from donors who are healthy. And therefore, this may minimize that manufacturing turnaround time. So we're involved with one of these allogeneic CAR T trials. And lastly, our cell therapy lab has the ability to grow some, do some homegrown CAR T development. I show a picture of a, it's like a bread box of CAR T machine where you stick in all the components of CAR T and out pops a fresh loaf of bread, but instead here what we see is a CAR T that can be developed at point of care and homegrown without requiring the use of manufacturing offsite. So very exciting advances in CAR T. Just a glimpse into what we're seeing. Next slide. And then on the left, I've talked largely about BCMA as the major target for CAR T and myeloma, but there are numerous targets that are being evaluated and numerous different types of CAR T constructs. On the right image, which may include using more than one CAR at the same time, targeting different markers, or maybe having two receptors on the same CAR construct, or having a switch that allows one to turn off the activation of T cells in the setting of excessive toxicity. So these are all very smart CAR Ts that are emerging and leading to a very fulsome research arena for CAR T. We are also moving beyond malignant heme, not just myeloma, lymphoma, or leukemia, but into solid tumors as well. And we'll start to see CAR T cell therapy combined with other immunotherapies and allow us to personalize the type of immunotherapy for individual patients. Next slide. So lastly, just as a summary, I just want to reiterate that cell therapy is a cornerstone treatment modality in myeloma for transplant and with CAR T moving into the future. We recognize, however, there are many challenges associated with cell therapy. It's demanding for patients, resource intensive, there's specialized expertise, and this is a very rapid pace of development in the research arena. And there are therefore many impactful opportunities for philanthropy to support our research, clinical care, as well as education and many other areas. So thanks very much.
Thank you so much for that update, Chris. Very insightful. And I'll just remind everybody who's listening in, if you want to answer your questions, we will have an open question answer period. If any of the technology is flying over your heads, we can certainly clarify that or take any of your other questions later on. But what we'd like to do now is to enter into a little bit of a conversation and discussion with myself and Donna and Chris. And anybody in the foundation who wants to join in is, of course, welcome. So let me start off asking both of you, just to let the audience get to know you a little bit. Donna, what inspired you to get involved in myeloma research in the first place?
Well, I started out as a leukemia and allogeneic transplant person when that was the hot item back many years ago. And then when autologous transplants became an effective treatment, and also myeloma had no advances in decades, the two met. And really, I wasn't sure what you would mention as the two main developments for myeloma. You said thalidomide and CAR T cells. I might have said autologous stem cell transplant and CAR T cells, but stem cell transplant was really the first treatment that made a difference for those patients with myeloma that could have it. And before we had access to maintenance, really could produce an unmaintained remission that would last 1.5 years, maybe 2 years. So this was a meeting of two different areas that then took off with thalidomide and its congeners, and is really not stopped in its development and momentum since that time.
Yes, I sort of remember the early days. Don, you were in Vancouver then. We were in competition to see who could do the first myeloma transplants, but it was healthy competition.
I think you beat us by a few months.
I wasn't going to go there, Donna, but since you mentioned it, I was still in Boston, actually. We sort of did it remotely. Chris, what about you? What inspired you to study myeloma?
Well, I started off my fellowship as a transplant fellow at Princess Margaret. And Keith, at that time you were on staff, and it was, you know, you used to ask the fellows to participate in your myeloma clinic, and it wasn't particularly popular because we didn't have very good treatments, apart from just, you know, the foray into transplant. And so I think I have to agree with you that thalidomide was so impactful for me, because I started working in your clinic and I would put a patient on thalidomide and see responses within the first month that were not really seen with any other modality of therapy to that point. So this made it super exciting. And then subsequent to that, we got Revlimid, pomalidomide, and all these, and bortezomib. So an amazing array of new drugs that allowed us to see gratifyingly immediate responses, like very rapid responses. And I think that really turned me onto the area of myeloma. And myeloma has been a really fulfilling area for not just patients, obviously, but for those of us who treat myeloma in the last 20 years. So it's so gratifying to see the survival of patients extending dramatically. I mean, you can't belittle the amount of research and advancements made in all arenas within the myeloma treatment landscape.
And the beautiful thing is we're not done, right? Every year brings a new, we'll hear about that in a minute, actually, because that's the exciting things. Yeah, I was just thinking as we talked about thalidomide that one of the inspirational things for me was the first patient I treated with thalidomide who was really sick. He was basically confined to bed, and he picked up his bed and walked. He came in, his nose stopped bleeding. He got up and led a pretty good quality of life for a number of years afterwards. And he actually gave me a little statue. He brought it back from Uganda, where his family was from. I still treasure that. And I was just wondering if you have also find inspiration from your patients, and what is about your patients that inspire you, Donna?
Oh gosh, I think we are privy to people in the fight of their lives. And I think being able to offer hope and optimism based on effective treatments is incredibly gratifying. And now we have, until we can cure this disease, we rely on sequential treatments to get as much time as we can out of each line of treatment. And I think one of the most thrilling aspects has been when all the funded options are done, you can open the door, and then there are these immunotherapies that are not mutually exclusive. One of the points I was trying to get through, this may have been when the video went down, was you can have a bite with one target that quits working. You can have, I didn't talk about a derivative of lenalidomide orally, then you can go to another bite, then you can go to a CAR T. It's like you open the door and there's a whole new sequence that you can run through, instead of stopping when all the funded drugs are used up. And these drugs are within the realm of being becoming standard treatments in the foreseeable future. So I think that part with taking your patient through that journey and having those doors open has been just incredibly rewarding.
Well, thank you. Chris, your thoughts on this? What inspires you from what inspiration do you derive from your patients or your colleagues?
Well, you know, when I started my career, we didn't have the array of therapeutic options we have now. So the relationship you develop with your patients, unfortunately, tends to be quite short. And over the last 20 years since I've been at Princess Margaret, I now have patients who I've known for literally 20 years, who are still living great quality of life. And with some of these new therapies, they're not on very much active treatment. Their symptoms are minimized. Their quality of life is actually very good. And so I am very much motivated by patients who have lived a long and good quality life with a very collaborative relationship with myself. It's really been an honor to actually care for patients for so long.
This is one of the joys of myeloma. You'd say, how can you enjoy being a cancer doctor? But our patients do live so long nowadays that you get to know them and their families, and that is one of the pleasures, I think, of doing our clinic. I was quite astonished when I came back after being away for 15 years, how many of my original patients were still coming to clinic. It was quite exciting to see, actually, which raises the question, I hate to use the C word, but cure. Donna, do you think we're curing patients today?
Well, I was taught the definition of cure is a patient lives their full life. And you don't know that till you see your patients live their full life. I think we're getting closer. I'm not certain we can say that, and I'm very, very cautious. I think we're getting closer with the tools we have now.
I saw recently that if you're over 75 at least, your life expectancy, the diagnosis of myeloma is very similar to an age-matched population of the same age. So that is essentially functional cure, is it not?
One could argue that. I'm just very, very cautious, but I'm very optimistic too.
So Donna, you just heard, I'm going to keep challenging you on this. You just heard of all these patients we've had alive for 20 years. Now, true, they still have myeloma, but I guess in terms of, going back to their pre-existing lives and having some kind of functional cure, if you want to call it that.
I think that is a very intriguing observation, since we know myeloma patients have these 5 to 7 subclones, and we see that all the time. They may have had vicious myeloma at the beginning, but what recurs after transplant is a very sleepy clone. It's almost like MGUS, and people can live with MGUS their whole natural life. So I think there are a variety of ways to look at the concept of functional cure.
Okay, I want to fast forward now, and we're getting some questions coming in about the future, and that's what many of our patients want to hear about. So, Chris, what's the most exciting, game-changing thing that's happening right now? I think you're going to tell me it's CAR T-cell therapy.
Yes.
I guess let me preface
Well,
let me add to that and say, when will it be available for Canadians?
well, that's a challenge, because I think what I hope to have relayed was the amazing efficacy of a single infusion of CAR Ts, but the huge effort and expense and resources required for the treatment of one patient. So this is not an easy therapeutic modality to roll out to large numbers in a publicly funded healthcare system. So big question is, when are we going to be able to access it? We do have limitations in terms of funding. I think what we need to see still is more mature data on what subsets of patients are going to benefit the most from this type of treatment, and not just use it as a big blanket treatment for everyone, and pick and choose, personalize the immunotherapy. Maybe one patient would be best off getting a bite followed by another bite, as opposed to perhaps a younger patient with a certain subset of high-risk myeloma should get the CAR T. So I think we have yet to learn where to place these different immunotherapies. I really don't think CAR T is going to be something that will be available to every single myeloma patient, nor should it. I don't really think it would be. It will be a necessary step for all patients.
And just for the audience, the CAR T probably costs close to $500,000 per patient. That's the downside. The upside is, we've talked about before, is they don't get any more therapy after that. So there's all the savings downstream and costs over the next couple of years. We don't yet know how many people will stay in remission for long periods of time. It's still too fresh. Donna, what are the barriers at Princess Margaret if you to look forward and say, where do we need to invest, and what were the barriers to our program continuing to be a world leader?
Oh, I think as you've highlighted, we need staff to do this. We need laboratory capabilities not to get these developments out of the lab into the clinics. And we need the coordinators, the nurses, we need the space. So I think we're pretty good with doing well with what we have, but I think if we had a larger day hospital, we could do more transplants faster. We could get the CAR T and bites done as outpatient faster. So I think there are a multitude of areas. I think our nurse practitioners have expanded our ability to take care of these complex patients. It's not good for patients getting complicated treatment to have to come and see us every week in clinic with the wait times and then have a treatment assigned to them. They do better with treatment on the spot by capable individuals. So I think any of those areas is rich for further development.
Yeah, and as Donna and Chris know, but maybe some of the audience don't, the Princess Margaret Cancer Centre has focused on 4 areas that we think we can excel in. One is the early detection, which the myeloma program is doing through molecular testing and liquid biopsy to replace bone marrows, and to look for disease very early before patients become symptomatic. The second we call beyond chemotherapy. That's getting rid of old-fashioned chemotherapy, including transplant one day, Donna. And that will be replaced, we think, with, I just saw the term, that there's a new class of doctors, immunologic interventionists. I anoint Chris Chen as our first immunologic interventionist, for example. Our third area is digital intelligence. So how do we move care out of the hospital into the home using technology? How do we take our digitized x-rays, our digitized pathology, our clinical databases, and apply machine learning and artificial intelligence to improve care? And the last one is the experience of our staff and our patients, the cancer experience, and how do we make that as efficient and seamless as possible. Chris, I'm going to give the last word to you before I hand over to Greg to introduce our guest. How do you measure success? What would you say is, when you look at our progress year over year, what do you think the metrics of success are and should be?
Well, there are measures of success at an individual level and at an institutional level. When we look at how, one measure is simply, can we provide proper care to the patients who need it? Can we provide care that is based on evidence? Can we provide leading edge care to those who, as I said, who need it? And so part of our metrics revolve around simply volumes, knowing what percentage of patients have myeloma in this first, second, third line and require treatment. Can we provide it to them? So it's an easy metric just to say we've, the incidence or the prevalence of myeloma is so-and-so, and we can provide appropriate care. But at an individual level, it's more, how can we impact the patient's quality of life? So as we've learned with trials, we are incorporating much more in the way of patient-reported outcomes, quality of care measurements. We don't want just to treat a lot of patients and necessarily just to have them live a few months longer. We want them to have good quality of life. So we need to always, with our trials, incorporate these subjective measures of success that if you were to even approach a single patient, we could evaluate that as a measure of success and not necessarily have to state that we've provided a large volume of care. So it's a bit tricky as a clinician to be able to clearly state how successful we are with care. As I said, especially in a publicly funded healthcare system where we may not have access to what we think is the absolutely best treatment. And hence we need philanthropy to help support driving forward the level of care for all patients that we may not be able to provide with government funding. So it is.
Sorry, go ahead. Sorry to cut you off.
No, that's okay. I'm starting to ramble a bit because it is a bit of a difficult.
Well, I would say that to me, the success is measured in, are we contributing to advancement of care around the world? And then a little bit of, are we making sure our patients have access to the most up-to-date current therapies, which essentially in Canada we have to do through our clinical trials program. So if that is advancing, we're advancing as well. And I think with that, I want to thank Donna and Chris for their time today. They're going to be around for the audience Q&A in a few minutes. And I'm going to hand it back to Greg, who's going to introduce Sass as our guest for the day.
Thank you, Keith. I have the privilege of working with many of you supporters who give direct donations to the myeloma program, whether through Gifts in Your Lifetime or Gifts in the Will. And however you give, and many of you are current supporters, you are contributing to the kind of progress that you've heard about today, enabling our brilliant colleagues to make this work possible. So thank you so much for that. One of the ways that the myeloma program is supported is through our 5K walk in the Journey to Conquer Cancer. And I just want to talk a little bit about that. So the Journey to Conquer Cancer, which is a run or walk event, 5K, started in 2012 and has raised almost $11 million for cancer research at Princess Margaret since its inception. And it offers 5K, 3K, 1K options, relatively easy options for participants. many of whom are grateful patients, family members, friends, kids, even dogs, all walking together to raise money for whatever cancer is closest to their heart. And this year, in 2022, we're thrilled to be bringing back our live and in-person event to Young Dundas Square, where supporters are going to be able to come together to have a very powerful experience, walking alongside many of our researchers, our clinicians, and also passing by Princess Margaret, which will bring our patients very much to mind. And we have many tremendous teams participating in the 5K, but the most successful team over the course of time has been the Multiple Myeloma M Moving Together Toward the Cure team, which is a group of extremely dedicated individuals who raise money for multiple myeloma research via the Molly and David Bloom Chair in Multiple Myeloma Research at Princess Margaret, which was established by Molly and David Bloom, who have been incredible supporters of the program. Molly is still with us. David passed away, leaving an incredible legacy. So this group of people comes together every year in this multiple myeloma team, and typically with a target of at least $500,000 to raise money for myeloma research. And to date, the group has raised almost $9 million since its inception. And so we know that many of you who are on the webinar today have been part of this in the past, and we hope that you'll be able to join again. This year's event will be on September 11th, and you can either join in person or virtually, and you can go to the Journey to Conquer Cancer website to register if you haven't already. So at this point, I'd like to introduce to you one of the longtime participants in the Journey, the myeloma event, Sass Khazzam. Sass has been actively involved in multiple myeloma fundraising and as a walk organizer since his wife Charmaine was diagnosed in June of 2015. And I'd now like to turn it over to Sass. Thank you for being with us today, and we look forward to hearing from you.
Thank you very much, Greg, for inviting me. I'd like to start off by thanking all major donors for their incredible generosity in funding research for multiple myeloma, as well as other cancers that they have so generously contributed to. Your generosity has helped and continues to help every family affected by cancer. Thank you. Charmaine received her tandem autologous stem cell transplant at Princess Margaret under the excellent care of Dr. Christine Chen. We are so fortunate to have some of the best doctors and researchers at Princess Margaret, and speaking with other patient families at the hospital during that period of time for transplants, especially those having had to relocate to Toronto to receive their transplants, helped me appreciate how fortunate we are to have Princess Margaret Hospital in our city. Obviously, I don't need to echo what you've heard beforehand from all our doctors. It's quite an achievement with Princess Margaret. Two people have had a significant impact in my having gotten involved. Cindy Leader, whose husband Harley Ulster had multiple myeloma, started the MM5K walk, now approaching its 18th year. And she had also advocated to Health Canada to approve the Janssen drug Velcade or Tezomib. By the way, that was the best drug for Charmaine's diagnosis, which she continues to receive bi-weekly. Cindy also introduced me to David Bloom, whom, along with his wife Molly, created the Bloom Chair in Multiple Myeloma Research with Dr. Donna Reese as the chair holder. As unlucky as we were with Charmaine's having multiple myeloma, we were so fortunate to have such strong advocates supporting crucial drug approval and research. Our family has been very fortunate to receive such strong support for our fundraising efforts from friends, family, and business associates. No matter how small a donation, every dollar counts to what is needed for research. Every multiple myeloma patient today benefits from research funded in the past, and our family is committed to helping patients today and those that will be diagnosed in the future. My relationship with Princess Margaret grew even stronger thanks to Dr. Chen, who asked me to help out a new charity that was providing subsidized accommodations for patients relocating for treatment at the blood clinic. It reminded me of conversations I had with families during Charlene's treatment and the difficulty and sometimes financial burden they faced. From its humble start in 2018 with 135 room nights, Staywell Suites charity provided over 12,000 room nights in 2021 to patients at Toronto's major hospitals. 2,100 of those nights were for Princess Margaret patients. The Multiple Myeloma 5K walk has raised about $9 million in its 17 years for the Bloom Chair, and all for research into improving treatment with the eventual goal of a cure. Over 90% of the funds raised go directly to research. As we've heard, there have been more advances in multiple myeloma research these last few years than any other cancer. Doesn't matter how small a donation is, it all adds up to moving the needle towards a cure for multiple myeloma. Please support multiple myeloma research by donating to the Bloom Chair, joining the walk, and please, those of you who are prepared to help out, please join our committee come this September. Thank you Thank you very much.
Thank you, Sass, so much. And thank you for being one of the many, many faithful people who over many years have contributed to this program. It's really an incredibly inspiring group of donors who are partners with us. So thank you for being part of that, and thank you to all of you who are listening for being part of our team. So we have about 19 minutes left before our 2:30 close. So now we can turn to some of the questions. Keith has already highlighted some of the questions I think that participants have brought forward, but we have some time for a few more before my closing remarks. So maybe, Keith, I'll start off with one that I see here in the chat, and then feel free to also bring questions forward from the chat. So we hear a lot about technology in healthcare. We know that multiple myeloma patients are immunocompromised and vulnerable to potentially serious complications. How is Princess Margaret capitalizing on technology in this area, in multiple myeloma research and care?
Well, Greg, maybe I'll take a stab at that first. Obviously, well, two major things to point out. Our new treatments are profoundly effective at killing plasma cells. In fact, we almost completely wipe them out. This is great because all the myeloma goes away. The downside is we completely ruin the normal immune system in the process. And this has become a bit of a challenge as our treatments have got better. The immune compromised state and susceptibility to infection of our patients has increased to the point that we sometimes have to replace their immunoglobulin, the proteins that plasma cells make, artificially. And we have to more and more rely on antibiotic prophylaxis. And of course, the COVID pandemic has put this in stark profile of the risk that our patients are susceptible to. For example, 3 vaccinations can be wiped out by a transplant or a CAR T-cell therapy. One of the technologies we're therefore fortunate enough to be embracing now is there are antibody prophylactic measures one can use against future COVID-19 infection. Those are just rolling out now. And of course, the new drugs that are available for COVID. In fact, during this meeting, I just got an email from Dr. Trudell about one of our patients who's just developed COVID and will be referred immediately to get one of the new anti-COVID drugs to help her. Having said that, let me turn it to Donna and Chris for any other comments about dealing with the immune compromised state and how we might embrace technology. Maybe Chris, I'll start with you. You're our immunologist interventionist today.
Well, sure. I think a nice example of how we're trying to bring in technology is for our CAR T patients who we know remain quite immunocompromised for extended periods, months on end. We've been looking at developing remote monitoring. Currently, when patients are discharged from hospital, we give them an oxygen saturation measure, a blood pressure cuff, a thermometer, and tell them, we teach them how to monitor all the pertinent vitals that may be able to identify early complications, even a month or two out from CAR T. But we are looking at developing some remote monitoring technologies where those vital signs will be directly relayed to our center without having to have that human as the intervening person. And so there are many different options using smart watches or all sorts of apps you can even just get that are commercially available. So we've been looking at that to try to enhance the monitoring of these very immunosuppressed patients after their CAR T treatment. And it can be used also to expand to other populations that are quite vulnerable. Also looking at perhaps the amyloid patients. These are patients who are particularly vulnerable to heart failure, et cetera. Early detection of abnormal oxygenation or blood pressure may be able to identify who needs to be brought right into hospital for immediate attention.
Actually, Chris, now you mentioned, I should have noted that the issue of wearable technology at home was one of our grand challenge winners in the cancer center, was wearing these hexoskin vests that were developed for astronauts to monitor blood pressure, pulse, oxygenation, and temperature that patients wear at home, and to see if that was actually accurate and sensitive in predicting early infection or complications. Donna, your thoughts on this?
Yes, I have several. They take a step out from individual monitoring of a patient, but the infection problems we're seeing now actually are reminiscent to me of some of the old allogeneic stem cell transplant days, donor transplants, when you wipe out the recipient's immune system and give them the donor's immune system, which is imperfect as it fits in its new home. So I think there are lessons to be learned with that. And I think our prophylactic measures, our early detection, our education of other physicians that will eventually be involved are all keystones to make this reasonable. I think our virtual clinics really offer patients the chance for more frequent visits without the inconvenience and potential costs of coming downtown, parking, et cetera. And those are going to stay in some capacity when the pandemic is over. I think too, we are using our database to look at the incidence of infection in our trials across Canada that involve monoclonal antibodies and other immunosuppressives to characterize our incidence of infection and look for risk factors. So I think these different strategies, along with the ones that you and Chris have talked about in terms of patients being able to be monitored at home with various devices that involve technology, are going to help mitigate the risks of infection that are inherent in our new treatments.
Thank you, Greg. We should probably go on to the next question.
Sure. So here's a question. For myeloma patients with the aggressive form who are in remission, but there is a concern of a recurrence, will CAR T therapy be available to help prevent recurrence?
Chris?
Remains to be seen the role of CAR T in someone who's actually in remission. Currently, it is a modality of therapy for patients who have active disease. And so, can it be something to a modality that can consolidate somebody's remission? We don't know yet, but that is certainly an intriguing thought. There are approaches to incorporating other immunotherapies like the monoclonal antibodies in maintenance to prevent further relapse. Also, even the BiTE therapies, ongoing maintenance post CAR T or post cell therapy, may be fruitful, but it's a bit early yet to know where to place CAR T in that setting.
Yeah, I think it's CAR T was started and people have failed everything, but they're very quickly moving along with the other immune interventions earlier in the treatment. And the trial that we've just opened and have been putting patients on recently is newly diagnosed patients who get CAR T therapy within 6 months of being diagnosed. And the question there is can you eradicate whatever disease is left, even if you can't see very much of it at that time? So not waiting until the patient has failed everything else. But in terms of being funded to do the work by the government, it will only be for some years, I think, only available for those who have failed other treatments. Greg, we should maybe go to the next question.
Sure. So congratulations on the advances to date. If you could predict the future of treatment for multiple myeloma patients, what will it look like 5 years from now? Now, to a degree, you've already answered that, but maybe you can give a consolidated answer.
It's a bit of a broader question. So Donna, this is yours.
I think we will be seeing we will get rid of chemotherapy as predicted by Dr. Stewart and advocated by Dr. Stewart, and these treatments will be delivered very early, likely as an outpatient with more technology monitoring to enhance the patient's experience and quality of life.
Very, very practically, Donna, one of the big changes we're seeing recently is moving from 3 drugs at the beginning of treatment to 4. Do you think that will become a standard? And do you think we need to keep treating people forever, or there's a point where we can stop?
I think we're learning about which patients might be able to stop treatment with the new tools like MRD combined with PET and mass spec and some of these other tools. Again, myeloma is a very heterogeneous disease, so I don't think there'll be a one-size-fits-all for all patients. Some patients have very indolent disease, and some, as we've mentioned, have very aggressive disease and need more tools brought to it to try to tame it. But I think the goal would be limited treatment because patients always feel better off all drugs, even if in our efforts to try to make ongoing treatment acceptable and have a good quality of life. Will there be every patient that can come off treatment? I don't know, but I think there will be a proportion.
Thank you. I could tell you one thing, I meant to mention it earlier actually, but one thing that will change is that we need a new generation of physicians who are very astute in these new technologies. And some of us, Donna and I in particular, are not getting any younger. So I'm sort of excited to tell you that we have potential to hire 3 new physicians to Princess Margaret in the next sorry, somebody's making noise. I said next 6 to 12 months. And all 3 of them are actually all 3 of them are currently in the United States at major East Coast famous cancer centers, and we're quite excited to see all 3 of them join us over the next few months.
I think too, Keith, and all of you, one of the amazing things about Princess Margaret is that we have a multitude of experts in many different areas who collaborate with the myeloma team. So, for instance, people who understand computational biology and are able to help digest all those numbers and the patient information and analyze them. So that's certainly another strength of the Princess Margaret, I would say, Keith.
Yes, and I was thinking not only did we introduce conscious sedation this year, but we have an advanced pain clinic for people who are suffering pain, often from the bony fractures they receive from myeloma. We've introduced procedural clinic where actually devices and nerve blocks can be used. We've started a cancer thrombosis clinic to support patients who might experience a blood clot. And we have fantastic support from our colleagues in infectious disease, as we've talked about, in managing, particularly during the COVID pandemic. So there are a whole host of ancillary services that are required: radiology, placing lines, imaging, advancing PET scanning, and other tools that are completely integral. Our molecular genetics lab, our pathology colleagues, we couldn't run a comprehensive program without all of those supporting teams.
I would also add our wonderful interventional radiology for vertebral augmentation. I think our capabilities at UHN for literally rebuilding a spine without operation, just by the judicious injection of cement into various vertebral bodies. Wonderful for immediate relief of pain, and literally doing multiple compression fracture treatments at a time, which is unusual and really limited to centers with outstanding expertise, which we have.
Well, we're getting close to the end of our time, but maybe I will ask just one of the questions that's in the chat. Princess Margaret is clearly a leader in treating multiple myeloma patients. What do we need to do to accelerate progress? And are there any limiting factors, i.e. space or people? To a degree, we've answered this, but any further comments on that question?
Yeah, I think Donna sort of addressed this, that all of the above, but you know, honestly, I think we have a pretty good cohort of staff today. It's not, we have a great team. I think it's the sustaining that team, it's accessing the most advanced technologies, supporting our basic researchers, supporting clinical trials that we want to do without necessarily having pharma funding. You know, advancing liquid biopsy through a combination of basic research and clinical implementation. These are things that, as Canada's Cancer Centre, we want to be at the leading edge of, and I think we have the team in place. It's more the operational funding to make these things happen that is probably our biggest limitation.
Yes, yes, and certainly, I mean, that's probably a good comment to end our questions on. And certainly, you know, from my experience at Princess Margaret Foundation, I've been here for 21 years. The things that you just talked about, Keith, many of them have been and continue to be supported by our donors. And you know, I'm starting to repeat myself, but again, our donors are so crucial.
I think we'd all like to thank everybody who joined us today. We'd like to thank the Veras family for their recent gift, and all of you have contributed to the program over the years. It's without philanthropy, we wouldn't be the program that we are today. And I wanted to say, Sass, I love the picture of your family. That was fabulous. It reminds us why we do this.
Thank you. Thanks very much, Keith.
Yes.
Especially the dog.
Yes, Sass, that was wonderful to see that picture, because absolutely reminds us about why what we're doing is so important.
That's what it's all about.
Yeah. So let me just make a few closing remarks. Thank you all for participating in this webinar, more about our multiple myeloma program. I want to give a special thanks to Sass, again, for your wonderful remarks, to Keith and Donna and Chris for your expertise, for the careful and thoughtful way you explained things today, and for continuing to drive forward this work along with your colleagues at Princess Margaret. And thank you to our community of supporters, together working with all of us together, our donors, our patients, the people in the Cancer Center, the foundation. Together, we are making progress, as you've heard today, and we will continue to do that. So if you're registered for this session, which you are, you'll be receiving a link in a day or two so that you can view the webinar again. We've recorded it, and also a brief survey with an opportunity to respond if you have questions, or if you'd like to move forward a conversation regarding giving. There'll be an opportunity for that. So thank you all, and until next time, stay safe, and we look forward to seeing you again. Have a good rest of the afternoon. Bye-bye.
Thank you.
Thank you.
Thank you.
Thank you.